All right? So when we think about microbiology, we kind of break it up into two major groups. And they're not totally mutually exclusive, but they're two different facets of what different people who are microbiologists might study. All right, so one is a basic biological science, and so that's kind of understanding things like the metabolism of the organisms, or where they're growing, or something about their genomes, and looking at how they work as cellular organisms. And then applied biological sciences, of course, are how we can use microbiology for, kind of exploit it for our own benefit. All right, and so we'll look and see that there are many different places in the healthcare, agriculture, industry, different areas where microbiology has become very important. I'm not going to spend a lot of time on this. I'll kind of point it out. But it's kind of a good, I think, a good overview of some of the different important things that microorganisms do. And it talks a little bit about origins of microbes and how they're responsible for doing all the things that they do. They're the largest, in terms of mass, they're the largest group of organisms on the planet. A lot of times people who study this kind of thing will call Earth the planet of the microorganisms. And so understanding microbiology is really essential for us if we're going to live here on their planet. Okay, so some of the different areas where microbes influence our world around us are in the environment. I mentioned this just a second ago. The majority of the world's biomass is microbiological, right? And they carry out metabolisms that allow for us to live here. So they could live here without us, but we couldn't live here without them. They will change and make different nutrients available to us that we otherwise wouldn't be able to gain. They're responsible for oxygenating the atmosphere so that we can breathe and we're able to live here. And so they provide a lot of the nutrients, et cetera, that we need. Also, if you kind of look at more of a, maybe look at this a little bit more of an applied way, there are places where in agriculture, medicine, food industry, et cetera, that we have used microorganisms for our own benefit. All right. Now, going back to sort of this basic microbiology, what kinds of things do people look at when it comes to looking at basic microbiology? Well, here are 6 important characteristics of cellular life. All right, so this makes that microorganisms actually a very good model system for understanding how cellular organisms work, how cellular life works. And that is because they have a metabolism. We can study metabolic pathways. We can see how they're gaining their energy, and that allows for them to grow. And so really just about every, I'm trying to think if there's an exception to this, but all microorganisms have, or all cellular organisms have the potential, at least for metabolism. They're going to change their environment around them because they're taking in some kind of nutrient, they're changing that carbon source, whatever it is, and then they're going to have waste products. And so they're always changing the environment where they are. And this goes for organisms, not only those that are using those carbon compounds as a carbon and energy source, but also for phototrophic organisms. That is the ones that are using light as an energy source. They still need to grow, so they still need to take in carbon compounds, and then they still need to put them into the macromolecules that make up the cells. Right? And then another thing, not all cells will divide, but reproduction is a kind of an important aspect of cellular life. And this might be organismal if you're looking at things larger than an individual cell, or multicellular organisms, for example. But even for microorganisms, so the main thing we think about is, they're growing, all right? So if they're growing, they're taking in some energy source. They're using that energy to incorporate new carbon compounds to make the proteins, to make the fatty acids, to make all the different things that they need, nucleic acids, that are going to allow them to then have enough to make 2 cells. And then they're going to assemble and divide into 2 new cells. And we'll talk more about cell division here as we go along. Another thing that cellular organisms will often do, so this is not something that is always found in microbes, or always found in cellular organisms in general, but cellular differentiation. And this is especially something that we notice with multicellular organisms, and this is one of the reasons why using microorganisms, which are much simpler cellular constructs, as a model system to study how things work in the more complex types of organisms. All right, so cellular differentiation, if you consider when you are first conceived, you're one omnipotent or omnipotent cell, and then by the time you divide, eventually you're going to have liver cells, kidney cells, skin cells, all the different types of cellular cell types that are present. Right? Well, microorganisms do this too. You might not think about it as readily, but certainly one good example of this is the formation of an endospore. That spore is actually a cell. And so this cell started to go through a cell division that gave rise to 2 different types of cells. There's this one, which is we'll call mother cell. We'll see that we'll call it mother cell later. And then this cell, which is a very resistant cell type that is metabolically dormant. It has the potential for metabolism, as we pointed out up here, but it can go for a long period of time without growing. Right? But it's got all of the genetic material that's present in the cell. Right? So it's just been impacted in here. This cell, the mother cell, is like a mother. It wants to do everything it can for its spore, that it's a cellular partner here. But this is a terminal cell type. So this cell will lyse open and will not be around anymore. But this cell will continue on, and it has the same genetic information, the same type of cell as this one here. Another thing you might not think about that microorganisms do is communicate with each other. There's cellular communication going on. They're releasing chemicals that are being picked up by other microbes that affect their behavior in some way. Usually it's going to affect gene expression in the receiving cell, and it's going to allow for them to work together really kind of as a unit. A lot of times we think about microorganisms as being individual cells and single-cell organisms, and they are for the most part, but they also can work together in groups as kind of an overall organism in of themselves. Motility is not something we see in all the microorganisms, but many of them can move around. We'll talk about locomotion a little bit later. Flagella are a pretty common way that we see them moving through a liquid environment, but they're also able to move across surfaces, and we'll be looking at some of those different types of locomotion a little bit later. The other thing that is pretty typical of cellular organisms is that they change over time. Their genetics change. So here we have an ancestral cell, and then over several generations, and this is really meant to be kind of over an evolutionary period of time is what we're looking at here. They're going to change. So there's going to be mutations that get introduced. If those changes are tolerable for the organism, and in some cases are going to be beneficial for the organism, then you take on those new traits, and then over time you're going to have the generation of new species as things have changed over time. All right. So I want to point out a big difference here between this, what's happening here. It looks like these cells are different, right? They're different from these ancestral cells. Over here, we have kind of a similar idea, right? This cell is different than that cell. But what's the difference between cellular differentiation and evolution? Yes? Mutation, right? So here, for the most part, there may be a mutation, a change of some sort within this cell relative to this cell. But for the most part, it's the same genetic information. Right? There's been no mutation going on here. Over here, that's what gives rise to all these new phenotypes, all these new different shapes and physiological features that they have, is that they've changed their, the genetics has been changed, and that allows for them to grow in different ways. Right? Now, when it comes to applied microbiology, some of those examples are shown here. So there's some in agriculture, some in energy, food microbiology, a very fun topic, by the way. And after you take this class, you'll be able to take the food microbiology class. Disease, if you want to take pathogenic bacteriology, that also will be an option for you. And then also in biotechnology, there's a lot of use of microorganisms in that field. Okay. In agriculture, we have a couple of examples here. There are a lot of plants that depend upon microorganisms to fix the nitrogen. So here, this is the type of nitrogen that makes up most of the nitrogen that's in the atmosphere. 70% is nitrogen, but that's unusable by us. We can't use nitrogen right out of the atmosphere in order to make amino acids, nucleic acids, types of things that require a fixed nitrogen form. Also, other cycles. These are biogeochemical cycles here: nitrogen cycle, sulfur cycle. There's also carbon cycle, phosphorus cycle. Lots of different. You probably learned about that in your general biology courses. All right, but microorganisms can kind of push these cycles forward. Right? They're able to do some of the things that we can't do, like fixing nitrogen into ammonia that can then be oxidized to nitrate. And so these can be incorporated pretty easily by multicellular organisms, eukaryotes, and other non-fixing prokaryotic cells. And then here this shows an example of animal husbandry. We see the cow out in the field eating grass. Right? Well, that grass is mostly cellulose. Most of the carbon and the energy that's stored in that grass is unusable for the cow. The cow can't do anything with the cellulose, but there are microorganisms that can break down the cellulose, and ultimately it gets incorporated as animal proteins. And this is through microorganisms that are part of the flora that are living inside of the rumen of the cow. And then over here, we're going to cover a couple of the. I actually don't think we'll get to this in this course. Talking about the use. A couple other applied places here we have. I'll talk about this one kind of a little bit later. But food microbiology is really two big types of applied science. One is how do we keep spoilage organisms out of our food. Right, so all the different kinds of treatment we can do to lower the microbial count. We can sterilize our food if we wanted to. It wouldn't taste that great. And so we have to come up with kind of a balance between getting rid of all the microbes and then just reducing their populations so that we have longer shelf lives. And we'll talk about that a little bit later as well. There are food preservation. So this is one big aspect of food microbiology. And then the other, which I think is the funner sort of thing, is using microorganisms to make food. So fermented food. Fermentation, and we'll see a little bit later, is two major groups of organisms that are yeasts that can carry out ethanol production, and that ethanol is used to make things like alcoholic beverages, beers, shown here, but also the distilled spirit, so it's making the product that's going to be distilled. And then the other group are important for making things like cheeses, sausages, other types of. And then in diseases, with diseases, where we can use microorganisms to identify the particular causative agents of a particular disease, and then we can also exploit what we know about their physiology in order to prevent their growth. And if we can prevent their growth, and that's a type of treatment, or in the case of things like vaccines, where we can actually prevent their ability to colonize our bodies to give us the diseases in the first place. Right? And then we have biotechnology, and this is largely what they're talking about here is making recombinant types of proteins where we can produce things like human enzymes in microorganisms. And a good example of this that's been very successful over the years is insulin. So insulin, most of the insulin that type 1 diabetics take is insulin that is human insulin, but it's produced in E. coli, produced in bacterial cells. Okay, so speaking of diseases and speaking of microorganisms, I want to make a comparison. This is over a 100-year period from 1900 to 2000, and if we look, this is just in the United States, the major causes of death in around 1900, if you look at the numbers here, all the things in red that are shown are related to microorganisms, related to infectious disease. Right, so influenza, pneumonia, tuberculosis, and even things like diphtheria here. So half of the major causes of death at the turn of that century were due to infectious disease. And then 100 years later, things are much different. Now we're hurting ourselves, our diets, etc., and heart disease, and you can see that there are only 2 that are shown here that are in the top 10. And during the pandemic, which is a little after 2004, it did move up into the top 10, but I think it's actually out of the top 10 now. So Robert Hooke, back in the 1660s, he was a polymath, he was an inventor, he did all kinds of different things, not just in microbiology. He was able to make microscopes and use them to examine the growth of things, and he looked at mold growing on leather, and of course they didn't have photographs back then, although Robert Hooke did invent the iris diaphragm, which is used in shutters of modern cameras. But at the time he hadn't the whole camera. So he was able to, he made drawings of what he was able to see. He also noticed coit. He looked at things like a eukaryotic cell, much bigger, and this is where we came up with the term, or he came up with the term cell. And a contemporary of his, also in the 1660s, 70s, 80s, we have Antoine von Leeuwenhoek, and he was a Dutch scientist, and he also was a microbiologist. And that's because he kind of came up with this way of making a spherical type of lens that he was very cagey about telling people how to do it. And he had lots and lots of lenses that he would make and incorporate them into his microscopes. And then it was really Leeuwenhoek who found prokaryotic cells. And we'll talk about the differences between prokaryotic and eukaryotic cells coming up pretty soon. Okay, now, all right, but by the later 1800s, we have who we really would call the founder of the modern bacteriology, and that's Ferdinand Cohn. And so what Ferdinand Cohn did, and again, so the other thing I'd like you to know about some of this stuff is really sort of the relative timeline. All right, so you had Hooke and Leeuwenhoek, who were in the 1600s, and then it was not until the mid-1800s that we had Ferdinand Cohn coming out. So what is it that he provided? What were his big contributions? Well, one of them is that he established a journal of bacteriology. And we still have, it's gone through a lot of iterations, but there still is a journal of bacteriology. It's administered by the American Science for Microbiology now. All right, so he was instrumental in setting this up as a real science where people could publish about their findings. Also, he developed a lot of basic bacteriology practices. What you'll see if you're in, how many people are taking the lab? Okay, so most of you are probably taking the lab. So you'll be seeing some of these practices. I don't want to confuse this with aseptic technique, which you'll be learning about how to do things without contaminating anything. But the best they had at the time were some of these basic bacteriological practices that he had for cleanliness and not contaminating things as best as he was able to do. All right. He also developed a classification scheme for bacteria. Now, this might not sound like such a big deal, but if you look at microorganisms, as many of you will be doing pretty soon in the microscopes, they all look kind of the same. Not all of them, but many of them are just rod-shaped or spherical cells, and you can't really tell the differences between different species just kind of looking at them there. You have to know something about what they look for, or what Cohn looked for, where differences in the physiology of the organisms. What kind of pathway? What could it use as carbon source? Or what could it grow on? What might be inhibitory to its growth. And so he was able to start to recognize here, we've got different species of organisms here, and we can actually categorize them and make a classification scheme here. The other thing that he was noted for is he's the first person to describe the life cycle of the endospore forming organisms. And this is really Bacillus, and I'll talk about this, we'll go into some more detail later. Bacillus and Clostridium are two genera that you should be familiar with. And Bacillus and Clostridia, the biggest difference between them is that Clostridia grow anaerobically, Bacillus grow in the presence of oxygen. But another person whose name you probably recognize, Louis Pasteur, he was really started out more as a biochemist than a microbiologist, and he was hired by the wine industry, and so he was able to look at the differences between the types of organisms that were found in wine. And one of the big problems was spoilage of wine. It starts to taste like vinegar, or it would become contaminated and beyond drinkability. And so he recognized the role of lactic acid bacteria in their fermentation in making these acid byproducts. And that's actually not something good in the case of wine. And then also the yeast that are present there, you recognize that they were there to do fermentation that had a different pathway, a different byproduct, and instead of being an acid or an acid byproduct, like you saw with the microbes here, the lactic acid, they were producing ethanol instead. And that was important for the wine to have the flavor that it has and have its purpose, right? So this was one of his a couple of the things that he did early on that kind of made him famous. Also, he helped to disprove spontaneous generation. I'll talk about that in the next slide. And then the other thing is he was an early adopter of understanding how vaccines work. So immunology was in its infancy at this time, and we didn't really understand a lot about it. We still have a lot to learn about immunology. But vaccines, it was recognized that if you used parts or if you used some aspect of an infectious disease organism and used it to inoculate into someone, if it didn't cause the disease, it usually would help them be resistant to the disease. And so he developed vaccines to rabies, chicken cholera, which would not to be confused with cholera that we're familiar with, and things like anthrax. So even early on, he was able to come up with some vaccines that were useful in preventing the spread of infectious disease over time. All right, so I mentioned this thing about this proof of spontaneous generation, and this kind of just outlines the steps that he used to do that. I think you kind of have an understanding of this. So if you took some non-sterile liquid, and this non-sterile liquid is a hay infusion. So you take hay and grasses and boil them, it generates what we call hay infusion, and it's actually got a lot of carbohydrates in it and a lot of nutrients that organisms like, right? And if you boil it, here it's non-sterile, we put it in the flask. We also want to talk about Robert Koch. So Robert Koch is one of my favorite guys for I don't know why, but he was a country doctor in Germany, and he was quite fastidious. He was a really science-oriented, sort of a researcher-oriented doctor, and he came up with methods for doing pure culture of microorganisms. So this is where aseptic technique really came into play. All right, and he also came up with postulates for determination of the causative agents of disease from microorganisms. All right, so he's really kind of the first person to say, look, this organism causes this disease. It always used to be, oh, there's some kind of contagion, right? There's some kind of germ that's associated here, and they knew about communicable diseases, but it wasn't really understood that there are specific microorganisms that cause specific diseases. Right? And he actually used his postulates to discover the cause of tuberculosis in the late 1800s. And so he came up with this idea that, first of all, if you have a diseased animal and a healthy animal, and we know that this is not 100% true all the time now, but he was kind of lucky in the examples that he went through. But what he found was that the diseased animal, you should be able to isolate organisms from that diseased animal and that are not present within the healthy animal. And so you've got something that's growing here, so this is a suspected pathogen. And I guess they're just really looking at a blood sample in this case. Right, so this is when he really got sick and died from things a little bit more easily. So again, with his ability to isolate pure cultures, he was able to grow. One of the things he said you have to do is be able to grow the microorganism in pure culture. You have to take that suspected pathogen or suspected pathogens, if there are more than one, and be able to grow them in pure culture by themselves. And that's what. Okay, the next guy I want to talk about is Martinus Pietering. We're going to talk about him until the end of the course. Okay, so really what we're going to do is talk about one of his contributions. And it's going to be something that we see come up time and time and time again. And this is the idea of enrichment culture or microbial selection. Really the same thing. Right? He came up with this idea of selecting for organisms. So what does that mean to select for organisms? Well, what it means is growing them under conditions where organisms that don't have a particular trait are going to be unable to grow, but those that do have a particular trait are going to be able to grow. And so you're selecting for them. So, or another example would be, what if you noticed down in the duck pond, you went down to the duck pond, and you said, there's really evidence of we're looking at chemicals, you said there's evidence of nitrogen fixation here. How can I figure out which microorganisms within the duck pond are fixing nitrogen? So you take a collection of some of the sludge out of the bottom of the pond, and you come back to the lab, and you say, okay, I'm going to want to figure out which organisms in here are able to fix nitrogen. What kinds of nitrogen into? That's what's not usable. So many organisms can take ammonia, and they can use it as a nitrogen source. They can tack it on to different compounds to make amino acids, and then they can oxidize it to nitrate if needed. Right? So they can bring it in, but the first thing that has to happen is to be fixed. The nitrogen has to go from into a usable form of nitrogen. Right? And there's only some subset of microorganisms that can do this. Right? We don't know if any eukaryotes do this. We can do it in a chemistry lab, but we can't do it, so that's how a lot of fertilizers are made. But you can't really do it in a living system. So what has to happen is you can take that sludge of microbes, all the different ones that are there, streak them out on a petri plate with a type of medium in it, and what would be the constitution of that medium? What kind of nitrogen would be in that medium? Well, nothing fixed. So if you don't put any fixed nitrogen, then essentially that means no nitrogen compound. no nitrogen-containing compounds that are salts, right? So then, the only organisms that are going to be able to grow are the ones that can take nitrogen out of the air, fix it into ammonia and then nitrates, etc., so that it can grow, right? So